Hello, hi. Welcome back to the part 4 of the lecture series of inheritance biology. In this lecture, we will be dealing about the linkage and recombination. In the past 3 lecture, we have dealt with the first lecture was about the basics and second was about the monohybrid cross and allelism. Then in the third lecture, we have dealt with the dihybrid cross. Now, we are going to deal with the linkage and the recombination. So, linkage and recombination. What is linkage? Right? Linkage means something has been linked, and recombination means a combination, which is that revised combination, right? A combination which is more revised and it is different from of the normal combination. So actually, I have mentioned what is recombination in the previous lecture. So today we will be dealing with linkage. So what do you mean by linkage? Linkage, it is the physical association of the genes on a chromosome. What? It is a physical association of a genes on a chromosome. So I am drawing a chromosome here. This is a chromosome, right? It is before replication. We know that after replication it will be having sister chromatids, right? So it is just consider a chromosome. So in this chromosome, we know that considering a gene here A and other gene here B and other gene here C. So we know that in a chromosome, the genes are fixed in their loci. Genes won't be moving. We know that in a chromosome, a genes will be fixed in a particular position. So, what is linkage? Linkage is the physical association. What do you mean by physical association? Physical association means we are here considering the distance between the genes, right? So, linkage is the physical association of the genes in a particular chromosome. We will be dealing it very deeply in the coming lecture. So, if you want to write the definition, you can write that we are dealing with the linkage. Linkage, it is the physical association of a gene on a chromosome. So what is the next heading is recombination. What is the recombination? Recombination, I actually know what you mean by recombination. It is a combination which is apart from that of the parental combination, which is a combination which is not like that of parental combination. So recombination is used to describe the non-parental combination. Okay, you can write it. Recombination is used to describe the non-parental combination. Okay, so we have dealt with the two definitions, what is linkage and what is recombination. Now just we are taking a condition here. Just write it down. Here is a chromosome. Okay. Here it is homologous chromosome. Okay. I am considering here gene A. Gene A. So the gene A will be same here, and its allele small a. This is known as loci of a gene, where the gene resides, the position of the gene, right? So I am taking here another gene, which is gene B. Gene B, its allele, right? Alternative form. So considering this cell is going to divide, it is going to undergo meiotic division. Okay, so in the process of meiosis, if no crossing over occur, what is crossing over or non-crossing over? We have dealt it in the cell cycle class. Okay, I will define it once more. First listen to this. So if there is a crossing over happening in this cell, okay, if a crossing over happens, what will be the combination here? So here first of all we are not considering crossing over. So what will be the condition? So after meiosis 1 and 2, this will be the condition of the cell. There will be 4 cell form having here A, A, B, 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 B. So there will be 4 cells, and this will be the 4 cells with haploid, which are haploid in condition. So you just think what will be the condition. Here the genotype is AB, here the genotype is AB, here the genotype is AB, here the genotype is AB. You look, it was the same as that of the parental condition, right? Here what is the combination? AABB. Yesterday we have done the cross. It is in the F1 generation. This is the condition. So after, without no crossing over, this will be the condition of the genotype, and it will be the parental condition. What were their parents? AB and AB. Look, this is same as that of the parental condition if there is no crossing over. So what happens if there is a crossing over? Okay. So we are going to check what happens if there is a crossing over. So if crossing over occur, crossing over, okay, CO. What happens in a crossing over? What is crossing over? Crossing over is the exchange of the genetical material of the non-sister chromatids of a homologous chromosome. These are homologous chromosome, and these two are the non-sister chromatids. You know, crossing over is a random process. Sometimes crossing over may occur in the prophase of the meiosis. Sometimes crossing over may not occur in the prophase. That is a random process. So a condition occurs then when we will discuss why there is no crossing over occurring and why there is crossing over occurring, because of the linkage distance. So first here we will be discussing, there will be, if there is a crossing over occur, what happens? If a crossing over occur, the condition will be like this. The crossing over will be like this. So what happens? I will describe it very detailed. So if a crossing over occur, we are considering 4 gametes will be formed, 4 cells will be formed, haploid cells will be formed, 1, 2, 3. After the end of the cell cycle, here A, here A, here B. First I will write this, it will be better. A, B here, and you know, now this A will be formed. It will be having, after crossing over, this genotype will be coming here. So small letter B, here small a and capital B. Here small a and small b. So I am writing down the genotypes. So the genotypes will be AB, capital A, small b, small a, capital B, and small a, small b. So there are 4 types of gametes being formed here. 4 types of cells have been formed here, and you just check the genotype. The genotype here is this genotype and this genotype are same as that of the parental condition, right? And you just check this genotype. These are apart, different from that of the parental condition. So they are recombinants. So there is a recombinant occurred due to the crossing over. You understand? I think you understand it, right? So a crossing over can cause a genetic variation. Why there is a genetic variation due to crossing over? Because in a crossing over, recombinant gametes will be formed with a different combination of alleles, right? A combination is different from that of the parental combination. So a non-parental combination will be formed due to the process of linkage or due to crossing over. Why this happens? We will be discussing it right now. Okay. So I think you have written it in your note. So now we are going to look much more deep in case of the linkage. What do you mean by linkage, and much more deep in case of linkage. So what is linkage? It is the physical association of a genes present on a chromosome. on the same chromosome, right? So linkage, we are going to describe linkage once more. Linkage, it is the physical association of a genes on a chromosome. What is the purpose of linkage? The purpose of the linkage is to find the distance between two genes or more than two genes in a same chromosome. Why I am saying same chromosome? Because a gene will be linked. We can define the linkage in only the same chromosome. I first draw a diagram ABC. We can only define the linkage in case of a same chromosome. So linkage can be of two types, or linkage can be of two types. We can say a linked gene or an unlinked gene. What do you mean by linked gene? Just consider a chromosome here. Chromosome. These are homologous chromosome. That means chromosome 1, and this is also chromosome 1. And I am considering two genes, gene A and gene B. Gene A and gene B. Or it will be better. These are homologous chromosome. So I am saying that this gene A and B are linked. So I am writing they are linked genes because they are present on the same chromosome. And there can be also a condition that I am drawing chromosome. This is chromosome 1, this is chromosome 1, this is chromosome 2, and this is chromosome 2. Gene A here, gene A here, gene B here, gene B here, or alternative form, anything. So in the second condition, I am saying that the genes are unlinked, which means the genes are present on different chromosome. Okay, this is the case of the linkage. So linkage can be, genes can be linked and also genes can be unlinked. Now there can be also another two condition also. In case of linked genes, there are also two conditions. I am going to draw the two condition in linked genes. I hope you are writing it everything. If you are having any type of doubts, you can ask in the comment box, because doubts may arise, because this is now this is a one-way communication. So doubt may arise and you can ask in the comment box or mail me. Okay, so we are now talking about linkage, linked genes. So linkage can be of two types. Linkage can be of two types. First is the complete linkage. So linkage can be complete linkage and also partial linkage. Partial linkage. What do you mean by complete linkage? So I am here drawing a chromosome. Two genes are in a very close proximity. Here, in a partial linkage, I am drawing two genes, gene A, gene B. These genes are in a very far proximity, or they are very far. Okay. So I am drawing its homologous chromosome, which with allele A, B. A, B. Okay. So in this form, or in complete linkage, you just think that if the two genes are together, the chance of crossing over in meiosis is less. Because I have drawn here, like I have drawn a line like this, a curve like this, because in the previous diagram, because there were a particular distance between two genes so that crossing over can occur. Here the chance of that linkage is very low. So we can say that here the genes are completely linked and the chance of crossing over is very less. If chance of crossing over is very less, the formation of non-parental types is very low. But it is different that in case of partial linkage. In partial linkage, the chance of crossing over is high because the genes are far in far distance. If the genes are far in distance, the chance of crossing over is high and the formation of the recombinant gametes are high. This is the definition of linkage. So linkage have connection with recombination. Okay? Recombination can be seen in linked genes and also unlinked genes. Yesterday I have defined how recombination occur in unlinked genes due to the possibility of the chromosomal orientation. Now here it is about the gene. We are talking about the gene in a linked chromosome. Linked genes. I think you are understanding it. So the chance there is high, here is low. So what will happen? So if this complete linkage undergo gametogenesis or mitosis, this gene both will be forming as a segregating as a unit. It will be forming AB. It will be also forming AB. That means parental type. The chance of these two genes as segregating as a unit, they will be going together during the formation of gametes because they are in a close proximity and the chance of chance of crossing over is very less. But there the chance of crossing over is high. So if crossing over occur there, then they can form the recombinant gametes. So if crossing over occur here, just if crossing over occur, what will happen? AB and AB will be formed apart from that of AB and AB. These are parental, these are non-parental. Here theoretically we can say 50% chance of occurrence of non-recombinants and occurrence of recombinant gametes are possible, but practically it has been seen that number of parental gametes are more than the number of recombinant gametes. So we have, I think you have understood it very carefully. This is the condition. So we can say that if the distance between two genes is very high, we can say that the chance of higher frequency or higher chance of formation of recombinant gametes are possible. Okay. I also mentioned that crossing over is a random process. Now, what about the recombination? Recombination and linkage are both linked together. So what is recombination? Recombination between the linked genes are used to locate the gene order. There is a particular order of the genes in a chromosome. So recombination can be used as a tool to identify or map the order, map the distance between two genes. Okay, and the degree of linkage is measured as a frequency of recombination. Yes, there is an equation also for finding the recombination frequency. Recombination frequency is the number of recombinants by total number of progeny into 100. This is the recombination frequency. If recombination frequency is high, which means the number of recombinant gametes are high, which means the genes are far away. If the recombination frequency is low, which means the number of recombinant gametes are low, which means the genes are together. And it is usually defined as a unit in centimorgan, or it is a percentage, or we can say it as a 1 map unit. If I say that the recombination frequency is 1 map unit, we should understand that the genes A and B are 1 map unit apart or 1 distance apart. This is the condition. Okay. Okay. Next, we will be dealing about the sex determination, right? So usually, before sex determination, there are 2 other topics in your textbook which are polygenic inheritance and also the pleiotropy. There are 2 other topics which are polygenic inheritance and pleiotropy. So what do you understand by polygenic inheritance? Polygenic inheritance. From the word itself, we can understand that polygenic, more than one gene or more genes are involved in inheritance pattern. Yes, that is the difference from Mendelian genetics and also polygenic inheritance. Mendelian inheritance and also polygenic inheritance. Look, I will say some difference between polygenic inheritance and also Mendel's inheritance. The first difference is that Mendel considered Mendel's inheritance was qualitative inheritance, which means he only looked about the qualitative character of a particular gene. But in case of polygenic inheritance, the polygenic inheritance deals with the quantitative character of a gene. Okay, so what is difference between qualitative and quantitative character? Please look. In Mendel's experiment, I'm just drawing a small axis, two axes here. In case of Mendel's experiment, the result was contrasting in nature, right? Either the plant was small or the plant was tall. He just looked about the quality, which means tall or dwarf. In Mendel's experiment, he didn't define what was the height of the plant. He just said that the plant was tall and the plant was dwarf. Okay. Okay. He didn't mention the intermediate stages or intermediate height between both the plants. We know that the nature is continuous in nature. The nature has continuous variation, right? We know that human beings are not having 6 feet only and not only for 3 feet only, but there are human beings which have a continuous or a variation in between these both two major length, right? There will be human beings with 5.7. Okay, so in Mendel's experiment, he only took the two conditions, either tall or dwarf. This is what you mean by qualitative nature. But in polygenic inheritance deal with quantitative character, which means, look, in polygenic inheritance we will be getting this type of polygenic inheritance deals with the intermediate characters also. This is the first point, first difference to be remembered. Okay, and the second point to be remembered is that in polygenic inheritance, in Mendel's inheritance, he didn't involve any environmental character or environmental factor. We know that we see in our environment or in our surroundings, environment have an impact on a particular organism. Look, I am growing an oak tree. We know that an oak tree will grow maximum to a height of 300 meters. That is the limit of the tree, 300 meters. After 300 meters, the tree can't grow. So, just consider a tree growing in a well-nutritious condition or a well-nutritious area in which it can absorb every mineral is readily available in the soil. And consider another oak tree where the minerals are not readily available and the water, sunlight is not readily available. We know that both these plants can grow up to 300 meters, but this plant will be growing maximum height because it has the maximum environmental condition. We can't say that this will grow to the maximum height. It also depends with other characters also, and this will be growing very retarded. It will be having a retarded growth because of the unavailability of the factor to grow, growth factors, right? So these are the two conditions. So in Mendel's experiment, he did not involve any environmental character. He just said that he did not involve any environmental character. So in polygenic inheritance, there is an involvement of environmental character. It is a quantitative study. And if we draw the graph, we can obtain a bell-shaped frequency. We will define it very deeply when we are doing questions, right? If we are getting questions from it, here, that portion we will be dealing it with it. So this is the condition of difference between Mendel's experiment, Mendel's inheritance and polygenic inheritance. In polygenic inheritance, a gene will be contributing an additive effect. We will say what is additive effect very soon. So the next portion is about the pleiotropy. In your textbook, the next thing mentioned is the pleiotropy. What is pleiotropy? Pleiotropy is a condition when a gene will be having more than one phenotypic effect. A gene will be having more than one phenotypic effect. This is known as pleiotropy. And in polygenic inheritance, more than one gene contribute to a particular character. More than one gene. So it is additive in nature. I will say one thing also about polygenic inheritance. Just consider a condition. Just consider a genotype AABB. This is the dominant form of the parent. This is also a recessive form of the parent. This is the recessive form of the parent. So we are considering two genes, okay, and we are considering two genes. This is a dihybrid cross in a polygenic inheritance. Just consider we are considering two genes in a polygenic inheritance. So I am just giving the condition that A in its dominant form can contribute. It's in its recessive form can't contribute. Just consider. And B in its dominant form can contribute. B in its recessive form can't contribute. So I am taking the melanin synthesis of a human being. What is melanin synthesis? Melanin is a particular chemical which is secreted by the skin to maintain the skin tone. If the melanin secretion is very low, which means the skin will be having a low tone or a light color. If the production is high, then it will be turning into a dark color. So these both two genes A and B contribute to the melanin synthesis. So A in its dominant form can contribute, B in its also dominant form can contribute the effect, melanin synthesis. So yesterday I have said that there is a shortcut method to find the F2 genotype. So I am writing the F2 genotype here, AA. Okay. So here both are dominant. So each gene can contribute a particular amount. So here 4 units of proteins or 4 units have been available because of the 4 units of the gene have been expressed. I said that A is the contributing allele. So what happens here? Here only 3 units will be formed. Here only 2 units. Here 3 units. Here what will happen? Here 2 units. Here only 1 unit. Here 2 unit. Here 1 unit. Here 0 unit of melanin. So this will be the unit synthesis of the melanin. So here 4 units of melanin will be produced. So the condition that the organism will be more darker. Here only no unit is produced. No unit means some contribution of the environment also will be contributed. That will we can discuss it later. Actually these things are not needed in textbooks. It is only mentioned in a small portion. So this is why that's the condition. Okay. So here 4 units of melanin will be produced. The organism will be more darker. Here the organism will be lighter. So there is a variation. We can see also intermediate form. We can see a organism with a lighter dark color also and much more darker color also. This is the condition in case of the melanin synthesis in human beings, and it is an example of the polygenic inheritance. Okay. So here each gene will have a contributing effect in its dominant form, and so it is also known as additive effect. So it is giving an additive effect in the phenotype. So this is the case of polygenic inheritance. Next I have also mentioned pleiotropy. Pleiotropy is a condition in which a gene have more than one phenotypic effect. I will just say it very quickly because if I keep on going explaining, the lecture will be not completed. So what is pleiotropy? I will say just very quickly. So pleiotropy. Just consider a situation. a product a reactant a substance A is converted to substance B with the help of a gene. Consider gene 1. And the B is converted into several other products C, D, E. B is converted into several other products with the help of gene 2. Okay? And there will be an intermediate product. Intermediate will be converted into the final product with the help of gene 3, 4, 5. Just consider it. Okay? Consider here there is an intermediate. So what is pleiotropy? If the product C is expressed, then consider that it will be contributing to skin color, D hair color or hair formation, and E like any voice change or sound production or any other phenotypic effect. So if you just consider, if gene 3 is mutated, the skin color can't be expressed. The organism may be white in tone or melanin because of the melanin production if gene 3 is mutated. If gene 4 is mutated, there will be a problem with the hair, right? And if gene 5 is mutated, there will be problem in the voice of the organism. Just consider if gene 2 was mutated, if gene 2 was mutated, it can't form the intermediate, so all these phenotypes can't be formed. So it will be leading to a larger issue. It is known as syndrome, right? Because this gene controls the other 3 genes. This is pleiotropic. This gene is pleiotropic gene. This effect or phenomena is known as pleiotropism or pleiotropy. So a gene 2 has a effect on more than one phenotype. This is known as pleiotropy. A good example of pleiotropy is phenylketonuria. Phenylketonuria is a good example of pleiotropy in which PAH gene gets mutated. Okay, in which a pyruvic acid, I think you know what is pyruvate, right? Pyruvate. I think I am not going much deep into it, that is not needed. Just understand phenylketonuria is an example. Okay. So next topic in your textbook is about the sex determination. We are going to determine the sex of both human beings and also honeybees. It is very easy topic, a small topic. So sex determination in human beings. We are talking about the genetic sex determination. We can also determine the sex of human beings phenotypically, right? We can check whether that organism is male or female by checking the phenotype. So we are talking here about the genetical sex determination. So we are going to study about the sex determination in human. A very easy topic. We know that in human beings, male is having XY, female is having XX. We know that male has a chromosome number, a diploid number of 23. We have a 23 set of chromosome, 23 pair of chromosome, which contribute to about 46 chromosomes, right? Okay. Sorry, here it is 46. We know that n is equal to 23. We have 23 set of chromosome. Okay. So in this 23 set of chromosome, we know that the last set XY is the sex chromosome or sex chromosome. The rest of the chromosome is known as allosomes. Sorry, autosomes. The sex chromosome are known as the allosomes. So in case of male, we are having a sex chromosome XY, and a female, in case of female, it is XX. So during the formation of gametes, we know that a sperm will be producing 50% of X chromosome. A sperm will be containing 50% of X and also 50% of Y. in 100 sperm produced, taking an example, if 100 sperms are produced, 50 sperms will be containing X and 50 sperms will be containing Y. Okay. Here what will be the condition? Here 100% will be forming X. 100% means 50x, which means 100% of the females will produce X. So what will happen in, I am writing it as X because we are going to fuse it. So after fusion, in case of fusion, we will be getting XX, XX, XY and XY, which means there is a, during a pregnancy, there is 50% chance that the progeny in the mother or the child in the mother inside her stomach or belly contains 50, there is 50% chance that it is a boy or it is a girl. 50% chance, like a coin. 50% chance that the boy can occur and 50% chance that the child will be a girl. So actually this 50% chance is donated from the father, because father is having two different types of gametes, mother is only having XX. So the chance that the progeny will be a daughter or a son is donated, or the determining factor is the father's Y, because father is having 50% X and 50% Y. I think you can understand it. But usually in our society earlier, usually girls were blamed for having a, usually the girls were blamed for having a daughter, because usually in our society it was considered that having a male was good, right? Not now, I am talking about the earlier condition. So usually the girls were blamed for having a lady because unaware, because of the lack of education, right? Here from our knowledge we can understand that father is the determining factor to whether the progeny will be a child or a daughter. So we are going fastly to the next. This is the case of sex determination in human beings. This is much more easier. There is much no need to give this topic as a prior importance, because the prior important chapter topics have been discussed earlier. Now we are going to study about the sex determination in honeybees. In honeybees, the condition is different apart from that of the human beings, because honeybees follow a haplodiploid sex determination. They follow haplodiploid sex determination, because in honeybees the male, the male or female is determined by the female parent. The female parent, which is the queen is the female parent. So queen determines, or queen is the determining factor to check whether the next progeny or the next generation should be male or next generation should be female. How? In case of a queen can produce two types of offspring. What are the two types of offspring? The first offspring is unfertilized eggs. Queen can produce unfertilized eggs. It can also produce fertilized eggs. The unfertilized eggs undergo division and forms the male honeybee. The fertilized eggs undergo division and forms the female honeybee. So here fertilization occur, so the condition will be 2n. Here no fertilization, so n. So here males will be n and females will be 2n. That means in case of males produce their sperm by the process of mitosis. but female produce their eggs by the process of meiosis. So female egg, usually female are diploid in condition, so while the gametogenesis they will be forming N. Males by the process of gametogenesis they can only form N. If the fusion occur, then it will be 2N. If the queen decides, I don't want to fertilize eggs, I need more males in the colony, then it will not fertilize, then it will be remain like this. So it will be remaining like this and it will be forming males. If fertilization occurs, then it will be forming females. This is the sex determination in honeybees. A very easy topic, I think you understood it. Males form their gametes or sperms by the process of mitosis, and females will be forming their eggs with the help of meiosis, which means they will be forming back to their haploid condition, the egg, but the male will be always in diploid condition. This is haplodiploid sex determination. It is also seen in certain wasps, but in wasps compared with honeybee it is different, just opposite. Diploid in case of male. I think you understood it. Okay, so actually this is the end of this lecture. In the next part we will be dealing about mutation. It will be in the next lecture. It is a very small lecture, mutation and pedigree analysis. So thank you. I hope you understood my lecture. If there is any correction in this lecture, or if there is any doubt, you can write down in the comment box or mail me. Okay, I hope you like my video. Thank you.